Hello, science fans. From galaxies above to the cells within, welcome to the Chromosome Carnival. Today, we journey deep inside to the instructions that build you, written in a microscopic language called DNA. So, what is cytogenetics? Cyto means cell, genetics means heredity. Together, it's the study of chromosomes. Think of it as visual genetics, seeing DNA's big picture packaging. Imagine a chromosome carnival. Rides that set traits like height or hair color. Some attractions even hold clues about health. Every living thing has chromosomes, the universal code of life. Cytogenetics gives us the map and magnifying glass to explore this inner world. Scientists learn this code to answer big questions. Here's our roadmap: basics of chromosomes, how we read them, why it matters, and a peek at the future. Ready to dive in? Let's go. Why you might share your parents' eyes or your grandparents' smile. Chromosomes. It's the universal programming language of life. Welcome to cytogenetics, science that's as spectacular as it is essential. First, meet the stars of the show, chromosomes. Chromosomes are tightly coiled DNA packages that hold genes. If genes are recipes, chromosomes are the cookbooks of you. They live in the nucleus of nearly all your cells. Humans have 46 chromosomes. 22 pairs of autosomes, plus a pair of sex chromosomes. XX typically female, XY typically male. One of each pair from mom, the other from dad. That's why you're a unique mix. Most cells are diploid. Gametes are haploid with 23. Stretch one cell's DNA and it's about 2 meters long. It wraps around histones, coils, and supercoils to fit and stay protected. During cell division, chromosomes ensure accurate copying and delivery of genetic info. Breaks or incorrect numbers can cause disease. Structure matters. Centromeres, arms, and banding patterns are our guide. They're the master organizers of the cell. What, where, how many, why packed, and what can go wrong. You've got the essentials. Now, how do scientists actually see chromosomes? A karyotype is an organized portrait of 46 chromosomes arranged by size. We catch them at metaphase when they're most visible. Common samples: blood, amniotic fluid, bone marrow, and skin biopsy. Cells are cultured and encouraged to divide. Colchicine arrests cells at metaphase. A hypotonic solution makes them swell, spreading chromosomes out. Dropping onto a slide helps separate them clearly. Stains reveal unique light and dark bands. Barcodes for each pair. Chromosomes are arranged by hand or software into a final karyotype. Fluorescence in situ, hybridization, FISH, lights up specific DNA regions. A fluorescent probe finds its match and glows under the microscope. It detects small deletions or duplications a karyotype can miss. If karyotype shows covers and FISH highlights a sentence, CMA scans every word. Chromosomal microarray compares your DNA to a reference, flagging tiny gains or losses with high resolution. Cytogenetics powers diagnosis in medicine: Down syndrome, extra chromosome 21, Turner syndrome, missing an X in females, Klinefelter syndrome, an extra X in males. Cancers often have big chromosome changes, deletions, duplications, or swaps. The Philadelphia chromosome, AT922 swap, marks chronic myeloid leukemia. Finding these changes refines diagnosis, prognosis, and treatment choices. In agriculture, chromosome tools help breed drought-hardy, higher-yield, more nutritious crops. In evolution, human chromosome 2 reveals an ancient fusion visible in our great ape cousins. Chromosome counts vary wildly. Fruit fly 8, dog 78, carp 104, and a fern can top 1,260. Gametes carry 23, embryos restore 46. Some people are mosaics, a patchwork of cell lines. Sperm carry X or Y. The Y's key gene triggers male development. From healing patients to feeding communities and tracing our roots, cytogenetics truly matters. How did modern cytogenetics begin? With a keen eye and a happy accident. Meet T.C. Shu, the father of modern cytogenetics. A lab slip led to hypotonic shock, swelling cells that beautifully spread chromosomes. Soon after, scientists proved humans have 46 chromosomes, 23 pairs, correcting decades of error. Recent achievements: non-invasive prenatal testing screens, fetal chromosomes from maternal blood, single-cell karyotyping profiles rare cells. Long-read sequencing and optical mapping reveal complex structural variants, and a telomere-to-telomere human genome assembly delivers a complete reference. As costs fall, chromosome-level analysis becomes routine in care. Oncology targets exact chromosomal changes for better outcomes with fewer side effects. Expect climate-smart crops and stronger conservation using chromosome insights. Significance? Cytogenetics connects DNA's structure to health, heredity, and biodiversity. Takeaways. Chromosomes organize DNA, labs can visualize and diagnose issues, and new tools see more than ever. We're just beginning to read this elegant code. If you learned something, please like, subscribe, and share. Stay curious, and see you at the next Chromosome Carnival.